biomarkers are essential for tailoring and designing the appropriate treatment for our patients with advanced cancers, not just for lung cancers, but for many, many cancers. Just focusing on lung cancers, we have a multitude of treatment options that are designed specifically and chosen specifically because of the tumor's biomarker. So what is a biomarker? Essentially, a biomarker is something that is unique and helps us to classify lung cancers. So way back when, before we used a lot of biomarkers, our pathologists, when we had a diagnosis of cancers, the only way we could classify these cancers was really based on how the cancer cells appeared under the microscope. And very basically, our pathologists would look under the microscope and tell us, this lung cancer is non-small cell lung cancer. And all that means is that it does not look like small cell lung cancer. And it was as basic as that. Our treatments were then divided based on whether or not the patient had non-small cell lung cancer, which comprises about 85% of all lung cancers, or whether the pathologist helped us classify them as small cell lung cancer. But in essence, aside from those two broad categories, we had no way of further classifying these lung cancers. And our treatments, consequently, were really based on whether or not the cancers fell into these two categories. Nowadays, we use more sophisticated testing and comprehensive testing of the cancer to better classify them. And that has translated into better treatments, more tailored treatments for our patients that are helping patients live much longer and potentially even be cured of their advanced cancer. So for lung cancer, our biomarkers we can test based on protein expression in the cancer cells. We can test the DNA to look for specific mutations in the DNA. We can also test the RNA, which is made by the cell from the DNA map, in order to look to see where the genetic alterations are in the cancer. And so based on whether or not the patient cancer has a specific biomarker in the DNA, RNA, or protein, we then use that information all together to design a treatment plan for the patient. All three ways of testing for biomarkers are essential to come up with the treatment plan. For example, for protein expression, one important biomarker that comes to mind is PD-L1. PD-L1 is a test that's done to look for protein expression by immunohistochemistry. And if the lung cancer highly expresses PD-L1, and does not have other genetic alterations in the DNA or RNA, then the treatment of choice involves an immunotherapy treatment because we know that that PD-L1 biomarker can predict for a higher chance of response to immunotherapy. In that same way, it's important to test for DNA mutations. So, for example, particularly pertinent in Asian populations is the presence of an EGFR mutation in lung cancer. About 20 to 30% of all lung cancers have a DNA mutation in EGFR. And it's particularly prevalent in patients who are of Asian descent and in Chinese patients. If the cancer has a DNA mutation in EGFR, then our treatment of choice is with a targeted therapy specifically that can inhibit EGFR mutated lung cancer. RNA fusions, also important to know about, because there are certain lung cancers that have alterations in the RNA, and that is important for choosing targeted therapies such as a RET inhibitor for RNA, RET fusion altered lung cancer. So all of these are essential. It's not choosing one over the other. All of them are important because they each look for a different biomarker. Now, insurance should cover all of these testing, and ideally we prefer to do comprehensive testing so that we can try to capture any or all of these alterations.